Hey Team Bio, welcome to your third screencast on organic compounds. Woohoo! Today is dive into proteins. So you probably have all heard of proteins before. I'm sure you have been told to eat enough protein in your diet. So you definitely have some information in your noggin about protein, and you might have some misinformation as well. So hopefully the screencast will be informative and maybe clear up some misconceptions. Okay, so proteins. All proteins contain carbon and hydrogen and oxygen and nitrogen. And some proteins contain sulfur, but all proteins contain these four different elements: carbon, hydrogen, oxygen, and nitrogen. So nitrogen, a little bit of a wild card, different than fats and lipids and carbohydrates. Okay, the main functions of proteins. Proteins can have structural functions. For example, keratin. This is what your hair and your nails are full of. So these are dead cells that have been packed chocolate block full of this protein keratin, which gives them the structural elements that they have. It's flexible but very durable. Okay, so proteins can play structural roles in cells. They can serve as hormones. So like fats and lipids, or lipids specifically, or I guess more generally, are cholesterols, which can be turned into sex hormones. There are a bunch of protein-based hormones. Some of them that you might have heard of are insulin. This is a hormone that's important for taking up glucose from the bloodstream into cells. Or human growth hormone. This is the hormone that stimulates growth, and if you took a bunch of it, you'd get super tall. Proteins also can be enzymes. And I don't think I've mentioned enzymes yet in this class, but they're really important to living things. They help speed up chemical reactions. We're going to spend a huge amount of time, probably most of the time that we spend talking about proteins, we'll be talking about enzyme structure and function. An example of an enzyme is salivary amylase. Salivary meaning found in saliva, and amylase is an enzyme that breaks up starches into glucose, because as you saw from the carbohydrate screencast, starch is just a polymer or a polysaccharide specifically of glucose monomers or glucose monosaccharides. So salivary amylase breaks down starch into more simple sugars. So if you take a cracker, like a stone wheat thin, and you just hold it in your mouth for a little while without chewing it and swallowing it, you'll notice over time it will taste more sweet. And this is because you are starting to break down those starch long chains of glucose into simple glucose sugars, and you perceive glucose to be much sweeter than you do starch. So it will start to taste sweeter over time and get all mushy. So chemical digestion begins in the mouth. Okay, a common misconception about proteins is that they are useful for energy, but they are not energy. They're actually the energy of last resort for your body. So basically, you'll only start to break down proteins in your body if you are starving. If you eat excess protein, if you have more protein in your diet than you need, which is the case for most of us, these proteins are what's called deaminated. This means the amino group is taken off of them. More on that later. And you pee it out. The other thing that happens to protein, if you consume excess protein, is that it's converted to carbs or fats. And you can enter cellular respiration. So it's converted to sugar or fat, and it helps power your body, or it can be stored. Most people think that they need protein for energy, but that's not true, actually. Your body is going to convert the excess protein that you eat to a different form, and then you're either going to store it or use it for energy in that different form, or pee out kind of the unnecessary nitrogen from that system. Okay, so I have a kind of a flow chart that gives us an analogy for monomers, dimers, and polymers of protein. Okay, so our example of a monomer is raw wool. So this is wool that's freshly shorn and I believe probably washed from a sheep. So the raw building blocks of proteins are called amino acids. And they all have the same common structure. They all start with a central carbon atom. And that carbon has 4 bonds to it. The most boring bond that it has is to a hydrogen. This is a kind of space filler bond because you know carbon likes to form 4 bonds in order to have a stable outer valence shell. But the bond to the right, or I guess it's a left, is a bond to the nitrogen. And that nitrogen, in turn, is bonded to 2 hydrogens. And the bond to the right is to a carbon, which is bonded to oxygen, hydrogen, and double bonded to a oxygen here. And then this I'm going to call R. More on that in a second. Okay, so we haven't quite gotten here in class yet, but we're going to be building different what are called functional groups. They are groups that are common kind of structural motifs or patterns that serve different purposes inside of living things. And this pattern or motif, this N bonded to 2 hydrogens, nitrogen bonded to 2 hydrogens, is called an amino group. And the pattern on the other side of the central carbon is called a carboxyl group. So we're going to be building these in class. The reason why this is called an amino acid is that this oxygen is pretty happy to give up this hydrogen in solution. I don't know if you remember this from methods, but what makes something an acid or makes something a base is acids are very willing to give up hydrogen, and bases readily accept extra hydrogens. So this is an amino acid because this group is an amino group and this carboxyl group is an acidic group. Okay, this last section that we have here, this R group, is called the variable group. And the variable group is variable. So this backbone of the amino acid, the amino group, the central carbon, and the carboxyl is always the same in every amino acid. They all have this common structure. But the R group, what the side chain, what here is represented by R, but could be a long string of carbons in a row. It could be carbons in a ring. We will build these variable groups later on in class. They're very different from one another, and they have different properties. So there are actually 20 unique R groups. And these 20, so there are 20 different amino acids because they vary based on their R group. And they're the same across all living things. So the same 20 amino acids that are in bacteria are the same 20 amino acids that are in you. So of these 20 unique R groups, 12 you can make in your body. And then 8 must be eaten in order to have them. You can't, there are 8 of them that you can't make. And we call these 8 essential amino acids because they have to be consumed, they have to be eaten in order to have them in your body. Okay, so the monomers of amino acids, they all look like this. They have this common backbone, and then they have a variable side chain. When we put 2 amino acids together, in a dimer, so if we have 1 amino acid plus another 1 amino acid, this is called a dipeptide. The bond that's formed between amino acids is called a peptide bond. So 2 amino acids is called a dipeptide. I know it's all this vocabulary. It's kind of annoying. I don't have a good kind of analogy for a dipeptide in my wool to sweater metaphor, but just stick with me. Okay. When we have a long chain of amino acids, so we take an amino acid and stick it to another one, we get a dipeptide, but if we add a third and a fourth and a fifth and a sixth and a seventh, we create a polymer. And we call a polymer of amino acids a polypeptide. So this is kind of similar to how if you take wool and you spin it into yarn, you get a long chain of yarn. However, this yarn in its ball formation, when it's just in this structure, you can't take this yarn and like drape it all over your body and expect to be very warm. In order to be warm from this ball of yarn, you have to knit it into a beautiful sweater. So the sweater is made from this yarn, but the pattern of the sweater is very important. It has to have a space for your head. It has to have sleeves for your arms. It has to fit your torso. So the reason why I'm making this metaphor is because if you take a long chain of amino acids and which we call a polypeptide, and you fold it in a special pattern. Then you get a functional protein. So this is also, I would say, a polymer. But in order to go from a polypeptide, just a long chain of amino acids, to a functional protein, this polypeptide needs to be folded very specifically. And if it's misfolded, then the protein will not function properly. And we'll talk in much more detail about the folding patterns that we need to use to turn a polypeptide into a functional protein later. But this is a basic introduction to proteins, their structure, and the fact that it's necessary to fold them in a specific shape in order to have them function properly. Okay, that's all.